Now, as I'm sure most of you know, human papillomavirus is a major cause of human cancer. Cervical cancer is by far and away the most predominant of this, where they account for about 100% of the cases. Papillomavirus has also caused a number of other anogenital tumours, and as well as a quite dramatically increasing amount of head and neck cancers. The WHO recently classified 13 different papillomavirus types as being cancer-causing of the cervix, and of course, by far and away the most important of these are these types HPV-16 and HPV-18, which account for about 70% of these tumours. The geographic distribution of cervical cancer is quite striking in that it really is a devastating problem in developing countries. This just shows you the distribution overlaid on the map of the ICGEB member states. But what's quite striking is that if you look here in Southeast Asia, the numbers are really devastating. It's the major cause of cancer-related death in women, and really it's reached epidemic proportions. I should also just like to mention that in Africa, the numbers might not seem so great, but there's also the usual issue here of underreporting, and the numbers are likely to be much higher than this. The virus, it's a small double-stranded DNA virus, and the unique feature and the critical feature about this virus is that its life cycle is intimately linked to the differentiation of the infected epithelium, whether that be at mucosal or cutaneous sites. It's believed to infect through microtraumas in the skin, where it infects the basal cells. And then as those cells differentiate and migrate up through the epithelium, we get coordinate expression of the different viral gene products such that we ultimately produce new infectious virus particles. Now, the key feature about this life cycle is that the viral DNA replication occurs in these mid-epithelial layers. As I'm sure you all know, these cells have exited the cell cycle, and there's no machinery available for replicating DNA. And so this is where the two viral oncoproteins, E6 and E7, come into play, and they kick these cells back into a kind of pseudo-S phase. They turn on cell DNA replication machinery such that you can replicate the viral DNA. Now, normally this whole process of viral life cycle takes something in the order of 2 to 3 weeks, after which time the infection is often cleared and the virus can become latent. What is clear, however, that in a small number of cases, you fail to clear these infections, resulting, and the virus remains persistent and readily detectable over a number of years. And this is the single biggest risk factor for ultimately progressing to the development of a malignancy. Now, during this malignant progression, what is also striking is that you get a very high and continuous expression of the two viral oncoproteins. And what is also clear is that this is really a dead end as far as the virus is concerned. You never get viral replication at this stage. And the reason for that is that during this malignant progression, the virus DNA frequently integrates into the host genome such that you lose a lot of the viral DNA coding capacity. So it's just not possible for the virus to replicate anymore. And all you're left with is high levels of the two oncoproteins, E6 and E7. And indeed, you can take cells derived from cervical tumours, such as HeLa cells, which have grown in tissue culture for over 50 years, and remove E6 and E7 expression from those cells, and they will enter apoptosis within a very short period of time. So this shows really very clearly that E6 and E7 continue to drive the development of these tumours, and they obviously also represent excellent targets for therapeutic intervention. Now, as I mentioned at the beginning, a small subset of viruses have been associated with the development of human cancer. In actual fact, there's over 150 different human papillomaviruses types have been described, and only a small proportion of those actually cause cancers. And this gives rise to this definition of high risk, which are cancer associated, and low risk types, which are predominantly only associated with benign lesions. And I think, one of the major questions that's been going on in the field for quite some time, is why do certain virus types such as HPV 6 and 11 only result in benign lesions, whereas we have types such as HPV 16 and 18 cause cancer. We've all done the experiments to look at the different interacting partners of the viral oncoproteins, and in many cases, it's fair to say that many of the cellular substrates are shared between these viral oncoproteins. And that's not surprising when we consider how closely related many of these virus types are. However, one of the exceptions, in my opinion, is the capacity of the E6 oncoproteins from the high-risk virus types to interact with cellular proteins that possess PDZ domains. And what is the reasoning behind this statement? Well, if we look at these different E6 proteins, all derived from cancer-causing papillomavirus types, and you look at this extreme C-terminal region of the E6 protein, you can see this very highly conserved stretch of 4 amino acids. This is a so-called PDZ binding motif. If we look in the low-risk papillomavirus types, these which are only associated with benign lesions, you can see that this motif is absent. So it can almost be considered to be a kind of molecular signature for potential to cause malignancy. This is just a schematic of the E6 protein to show you where this region lies in relation to everything else on E6. And one of the really nice things about this is that we can make mutations in this domain of E6 without affecting any of other E6 function. And so we can really ask questions about what does this set of interactions do with respect to the transforming capacity of the protein and also with respect to the viral life cycle. Unfortunately, we can't really do those sort of analyses with most of the other interacting partners of E6. And of course, within this, we have P53, BAC, E6AP, and so on. And that is because many of these sites of interaction are overlapping, and mutations in one region of E6 can have detrimental effects elsewhere. So it's very hard to pin down what many of these other interactions are doing with respect to viral transforming potential. So what about PDZ interactions and the biology? What do we know about it? Well, if we look in transgenic mice, we know that if E6 cannot interact with these proteins, it really inhibits dramatically its ability to cooperate with E7 to induce tumours. In tissue culture systems, a whole variety of assays have shown quite clearly that this set of interactions is important for the capacity of E6 to contribute towards transformation. And perhaps most importantly, and this is the key point, these interactions have evolved for the life cycle. It also is, if you make mutations within E6 in the context of the whole viral genome and leaving everything else intact, you see a great reduction in the number of cells that are capable of supporting viral replication. You also see a greatly reduced rates of viral DNA replication. And in actual fact, over time, you lose the viral episomes. So the virus cannot maintain itself within the cell without E6 having PDZ binding potential. Now, a whole panel of different PDZ domain-containing proteins have been described as E6 interacting partners. Many of these are classified as potential tumor suppressors. They're involved in regulating signaling complexes. But what I'm going to do for the sake of today's talk, I'm just going to focus on two of these, the two that were first identified, DLG or disc large and Scribble. These were first described in Drosophila, where they were classified as potential tumor suppressor proteins, and they're involved in processes regulating cell polarity. Why were we particularly interested in these targets? Well, as I say, they're involved with regulation of aspects of cellular homeostasis, breakdown of which can all be considered to be critical during the development of malignancy and the progression to metastasis. Now, they belong to this cell polarity regulatory module. This is just showing the model from Drosophila. It's a tripartite multi-macromolecular signaling complex where we have DLG and scribble define the basal-lateral component of the cell. We have the subapical region defined by the PAR complex, and the apical part of the cell defined by the crumbs. And the key point about this is that their locations are all strictly controlled by a series of mutually antagonistic interactions. And the main point to remember is that mislocalization or loss of expression of any single component of this complex will greatly perturb the functioning of the whole. And indeed, that's what E6 does. It's a very potent degrader of the dislodged protein. And this is the E6. You can see in the presence of 18E6, dislodged disappears. With 11E6, which doesn't have a PDZ binding site, it has no effect on DLG, whereas 16E6 can degrade DLG to some extent, but is never as good as 18. This was sort of a slightly strange observation, and when we go and do a simple interaction assay, the explanation is quite clear. 18E6 binds to DLG much more strongly than 16E6. When we go back to the sequence, the PDZ recognition motif on E6, the reason for the difference is not really that obvious. If you look here, there's a great degree of similarity between HPV16 and HPV18, except this last C-terminal amino acid, where we have a leucine on 16 and a valine on 18. And we did ask the question, is this single amino acid difference sufficient to explain the difference in the capacity of these two viral E6 proteins to target the DLG protein? And indeed, that is the case. If you look here, what we did was to just swap that last amino acid between the two proteins, and we look at DLG degradation here. You can see that with 16E6, it's quite weak. With 18E6, it's very efficient. You swap the last amino acid on 16 to make it look like 18, and it becomes a lot more efficient. In contrast, if you make 18E6 look like 16, it's essentially inhibited. The other striking thing about this set of analyses was with scribble, which is the other component of this cell polarity module, it's actually the other way around. So 16E6 preferentially targets scribble. So what this sort of seems to indicate is that we have two viral proteins targeting this cell polarity regulatory network through a very slight alteration in this last PDZ recognition motif, you can alter subtly which cellular substrate you're going to go for. And so this really prompted analysis to try and dissect the details of how E6 recognizes PDZ domains in more detail. And this was a wonderful collaboration with Xiaojing Chen in California, where we obtained these three crystal structures. The first one is showing you the E6 peptide bound to the MAGI1 PDZ1 domain. I'm not going to say anything about MAGI1 today, other than that it's the strongest bound PDZ domain substrate that we know for E6. There's this structure which shows you E6 bound to the PDZ2 domain of DLG, which we know is the natural target site of E6 in vivo. And this structure here, which is really a control, it's E6 bound to the PDZ3 domain of DLG, which it normally would not recognize in vivo. This is just an artificial in vitro generated interaction where you can push these associations quite hard and still nonetheless see the complex. And combining this with mutational analyses, what it told us was that for those domains that are really bona fide interaction sites of E6, we have residues within the PDZ binding motif outside the canonical recognition sites that contribute to recognition specificity, such that in this site here, we have residues in addition to the -1 and -3, which are the canonical interaction sites. We have additional contact points at the -2 and -5. And in this really much more strongly bound interaction, we have an additional contact point at -6. So what this tells us is that if we look at all these different E6 proteins, which are from the high-risk associated HPV types, you can see there's a lot of variation within these residues in these PDZ binding motifs. And it suggests that as we look at more disparate different virus types, we're going to see different types of PDZ recognitions taking place. What we can also do with this information is to do in silico screens to identify potentially inhibitory interfering compounds that might block the ability of E6 to interact with these proteins. And currently we're analysing a panel of these now on the effects on cells derived from cervical cancers. Obviously, these should turn out hopefully be useful reagents for studying how PDZ interactions are important for viral life cycle and cell transformation, and they also obviously have, we hope they may have some potential therapeutic potential. I'd just like to digress slightly at this point and just say a little word about rhesus papillomavirus. This is really probably the best model system for looking at cervical cancer in a natural host. The rhesus papillomavirus causes cervical cancer in rhesus macaques. It has many similarities with HPV 16 and 18, and it's sexually transmitted. If PDZ binding was going to be an important feature, you would have predicted that the E6 protein would also have a PDZ binding motif, but in actual fact it does not. There's nothing there that at all fits the consensus. What was striking when we were looking at this, however, if we look at the E7 protein, which is the other player in the drive towards malignancy, you can see there is in actual fact a perfectly conserved PDZ binding motif. And this, although very different from that which is seen in the HPV E6 proteins. We did proteomic analyses to look for which cellular proteins might interact with this, and we just asked a very simple question. In this analysis, which is the most preferred PDZ cellular substrate to be bound by these different viral oncoproteins? Well, 16E6 was Scribble, not surprisingly. 18E6, it was DLG. What we found with rhesus E7 was only one cellular PDZ domain containing protein was pulled down in this analysis, and that was a guy called PAR3. As I'm sure some of you remember, PAR3 is belonging to exactly the same polarity control network as DLG and Scribble, except that it defines the subapical region and is in close proximity to the tight junction. And indeed, perturbation of this complex is almost even more fundamental to the regulation of the whole pathway than is DLG and Scribble. So yeah, rhesus does actually interact with this protein. This is just a simple interaction assay, and you show here that the association is actually, it is PDZ dependent. And what was also very pleasing to see was that 18E6 really doesn't see this particular cellular protein. And that's not surprising when we consider the differences in the recognition motifs. What does rhesus E7 do to it? Again, like the E6 protein with its substrates, it actually results in a degree of protein degradation as well as relocalization. So again, it's perturbing the functioning of this signaling complex. So I think this sort of analysis was really quite exciting. It showed that really across evolution, papillomaviruses are targeting the Scribble pathway, albeit through two different mechanisms. HPV E6 is using PDZ recognition to target DLG or Scribble, whereas rhesus, it's using the same form of molecular recognition, but it's using a different viral protein and a different cellular substrate. So in the last sort of couple of minutes, I'd just like to sort of finish with some of our ideas about why does E6 target these proteins. Now, obviously, you might say, well, it's to cause cancer, but the answer has got to be a no to that. It might help cause cancer, but it's not why the virus does it. It's obviously got some critical role to play in the capacity of the virus to replicate. And does the virus need to alter cell polarity? Well, at this stage, I can't answer that. We really don't know. Is it to affect cell signaling and proliferation? And I think the answer to that is certainly yes. And so the way we're approaching this is to just essentially try and knock down DLG Scribble expression in keratinocytes and say, what does this do to cellular homeostasis under these conditions. If you look at cells that lack DLG, they actually grow in a very nice cobblestone morphology. They really don't look much different from the control cells. In contrast, if in the scribble deleted cells, you can see that you're already beginning to see some striking changes in cell morphology, and they've certainly lost a lot of this cobblestone morphology. In terms of proliferation, scribble depleted cells grow very rapidly. They form high degrees, they're very efficient at colony forming capacity. Whereas in contrast, DLG depleted cells actually grow very slowly. And this was really something of a surprise. It's something we were not expecting. If you look at the capacity of these cells to invade in matrigel, again, scribble knockdown cells are very invasive and very aggressive in these types of assays. Again, DLG removed cells, again, counterintuitive, really do not invade through a matrigel, in a matrigel system. What about the capacity of these cells to adhere? Again, scribble behaved as expected, very low adherent cells, but once again, DLG was opposite to what we were thinking. Again, we see here cells really aggregating very efficiently. So this led us to the last series of analyses, which to look at the effects of the loss of these two proteins upon the susceptibility of the cells to enter apoptosis, and in particular, anoikis, where cells are suspended in a substrate-free environment. And what we found there, most surprisingly, was that in the DLG knockdown cells, they're highly resistant to anoikis induction, whereas the scribble cells are very, very susceptible. So what this seems to show is that loss of scribble seems to be behaving as a classic tumor suppressor in human keratinocytes, whereas DLG has certain tumor suppressor characteristics in terms of its ability to regulate anoikis, but it may also have certain degree of proto-oncogenic potential. And I'd just like to summarize that PDZ targeting does seem to be specific for high-risk cancer-causing mucosal types. We think there's a large diversity in PDZ substrate targeting by the different HPV E6 proteins, and a common pathway of polarity and proliferation control is targeted by different papillomavirus types across evolution. And we can see that loss of DLG appears to promote cell survival, whilst loss of DLG, scribble, is promoting proliferation and invasion. So I think what it tells us is that E6, by subtly altering how it targets scribble and DLG, can have differential effects upon cell proliferation, invasion, and apoptosis, ultimately fine-tuning conditions for viral replication. I'd just like to acknowledge the members of the lab who have been involved in some aspects of this work. Paolo Massimi, who's been doing the knockdown experiments, Miranda Thomas, who has been doing all the structural analyses, and Vyacheslav Tomich, who has been doing the research work, and of course a fantastic collaboration with Xiaojing Chen in California. Thank you very much.